All right, hello everyone. Today we are going to hear about the PhEval framework for benchmarking variant prioritization systems. And Yasmin from Queen Mary University is going to run us through how to set up a simple benchmark using the framework, and hopefully make it possible for you to kind of like set up your own benchmarks with your own projects as well. So thank you, Yasmin, for volunteering and taking the time to prepare this. I will, take it away from here, and look forward to everything. You're still muted.
You're muted.
You hear me now? Can you see my slides?
Yes, you can see
Okay.
your slides and hear you.
All right, thanks, Nico. So today I'm introducing you to PhEval. So this is a benchmarking framework for phenotype-based gene and variant prioritization tools. So the seminar today is called Introduction to PhEval and How to Run a Simple Benchmark, but don't let the word simple fool you. So benchmarking in this space is anything but trivial. And my goal is to walk you through why it's so important and how PhEval helps make this process reproducible, interpretable, and scalable to any size. So I'll show you how to actually use PhEval, not just what it is conceptually, but how to get it running in practice. And this should hopefully give you a strong foundation into understanding how it works and why we have it. So here's what I'm aiming to cover this session. So we'll start with a bit of the background on the problem space, specifically the challenges we face in rare disease diagnosis and the role of phenotype-driven prioritization. So this sets the frame for why we need PhEval. Then we'll talk a bit about benchmarking, why it's so important, and then from there I'll introduce you to PhEval, what it is, why we built it, and how it's structured. The next section is a bit more hands-on. We'll look at how PhEval is architectured internally and how it interfaces with several tools that we've implemented as runners like Xmiser and Gado. And then finally, I'll walk you through how to configure and run a benchmark, and I'll show you some real output from some time at the end for some questions and answers. So let's begin with the problem. So rare diseases affect over 400 million people worldwide, so with over 1,000 different conditions. And diagnosing these rare diseases is an incredibly complex and time-consuming process. The average time for diagnosis is from 4 to 5 years, and we often refer to this as a diagnostic odyssey. So the reason that they can be so difficult to diagnose is that around 80% of these have a genetic component, and this, along with the heterogeneity of phenotypes, so this is where the same disease might present differently in two patients, or the phenotypes of the condition may overlap with more common conditions, is what causes it to be such a big challenge. So in this space, there's been huge progress, largely because of next generation sequencing, so it's now accessible, but interpretation of this, the output is bottleneck. So the sequencing gives us tens of thousands of variants per patient, and the real challenge is prioritizing these to identify which ones might actually explain the phenotype of what we're seeing in the clinic. And what we'll often see is that clinicians don't have the time to sift through such a large amount of data to help diagnose the patient. So this is where phenotype-driven variant and gene prioritization algorithms come in. They combine clinical phenotypic features, which are often encoded as HPO terms and IDs, with the variant data to try and rank candidate genes and variants and give us like a list of what the potential candidates are for each patient. But the catch is, how do we know whether these tools are actually performing well? And that's what we'll explore later in the presentation. So there are a variety of algorithms out there that attempt to tackle this task. There are tools like Xmiser, Lyrical, and AI Marvel, and many others that combine genotypic and phenotypic data, or just the phenotypic data, to try and evaluate whether what are the likely causative variants, genes, or diseases towards the top of a ranked list. So ideally, in a clinical setting, what we set up for Xmiser is that we want the known entity to be within the top 5 ranks. So this limits the amount of time that clinicians are spending sifting through the results. So each of these tools, they differ in what kinds of data it uses in these reference databases. Some rely more heavily on known gene to disease associations, others leverage gene expression data or interaction networks, or also model organism and orthologs and phenotypes. They may also differ in how they interpret phenotypic similarity. So some may use through semantic similarity methods like Resnick or Jaccard scores. So in theory, these tools should help narrow down from thousands of variants to a small manageable set of candidates. But in practice, we see a lot of variation in the performance of these tools. So the image on the right is just a very general diagram of how these variant and gene prioritization tools may work. And then you can see at the end, we get this final prioritization, which could be genes, variants, or diseases. So the key problem is that we don't have a standard way to compare these tools. So if you've ever tried to evaluate one of a different tool or reproduce some results from a paper that has done like a wide benchmarking study, you may have found this problem yourself, that it's very difficult to reproduce the results that they report. So this brings me as to why benchmarking matters. So imagine you're building a new algorithm that's prioritize some variants or genes, and you run your tool on a data set and get a bunch of score. But what do you do now? How do you know if you've improved on what the state of the art tool is right now, or whether your results are even meaningful? So historically, there's been no standardized framework for benchmarking these type of tools. So as a result, people tend to use different data sets, and this makes comparisons difficult. And what I mean by data sets is the patients that they're using to benchmark these tools. Some of them are private, so we can't actually replicate the performance. For example, if someone did a private data set on your own tool and their performance is highly variable, less than what or what you predict your tool to perform, it's very difficult to kind of reproduce those results and see whether something's been configured wrong or those results are actually truthful and what like a weak point in your algorithm. Also, the metrics aren't always consistent. Some outlook on the results. So this doesn't provide a whole picture on how the algorithm works. Tool configurations also vary widely, and it's often unclear what version of a tool or what database snapshot it replied on. So these tools are often complex with many configurable settings, and these can be manipulated and could potentially be misconfigured for the use that it was intended for. And this could be because of a lack of understanding of how the tool works. And this is what can lead to the inaccurate results and variable performances reported in numerous studies. So these factors can lead to results that are non-reproducible, which is a critical issue, especially when these tools are used in diagnostic pipelines. So ensuring reproducibility and accuracy is vital for reliable diagnosis and ultimate better patient outcomes. So all of this makes progress hard to measure. So what we're trying to change that with is PhEval. So we're introducing this standardized framework, which will help enable these objective comparisons. So we can compare these tools across the same cases objectively, and we can track improvements over time across versions or configurations of a certain tool. And we could even understand how phenotypic features or ontology updates or even small algorithm tweaks may affect the performance. And ultimately, this helps translate into clinical utility more effectively, which for rare disease patients can mean faster and more accurate diagnoses. So that brings us to PhEval. So PhEval is a benchmarking framework, and it's designed specifically for phenotype-driven prioritization tools. And it's built to be modular and extensible, meaning you can plug in your own tools, run your own data sets, and get standardized outputs. So we designed it not just to support research, but to help push towards routine benchmarking. So tool developers or people who more in the back end and the database stuff can rapidly iterate, and users can make informed choices about what tools are suitable for their use case. So the PhEval paper was published last month, and it's in BMC in bioinformatics. So PhEval was designed to solve some of these persistent pain points in rare disease tool evaluation space. So this is just some of the key value propositions that we have of the tool. So we can begin with the standardization. So by normalizing the inputs, outputs, and the performance metrics that as an output, PhEval ensures that when you compare the tools, you're actually comparing like with like. So this is critical for fair and reproducible benchmarking. Secondly, again, it's reproducibility. So we're making sure that the results these algorithms generate can be trusted and consistently replicated. So what's key here is that when developers implement their own tools into PhEval, we can make sure that they are outlining the optimal settings or the right settings to actually run their tool and get the results that they would expect as well. Thirdly, PhEval helps with automation, so it involves less manual effort by automating several tasks. For example, so you have a set of patients in phenopacket form. You know your tool will represent the gene identifiers as in the namespace, but the phenopackets are in HGNC. There's like several PhEval utility tools that can map the HGNC identifiers in the phenopacket to Ensemble. Also, we'll run the whole process from start to finish with the tool, like with the which I'll go through in the architecture. And then finally, it performs statistics, which include both binary classification and ranking metrics. And this will help show the performance of an algorithm across all aspects and help pinpoint any potential weak points within your tool. And there are also more in-depth comparisons involved where you can review the performance case by case. So you can look at for each patient and each variant gene or disease, and you can see how rankings fluctuate between different configurations. So I will just walk you through some real-world scenarios where PhEval can be particularly useful. So one major use case is for comparing multiple tools. So if you want to know whether Xmiser or Gado or Lyrical, AI Marvel performs better on a set of rare disease cases, or even which version of a tool works best after about to set up an apples to apples comparison. Another use case is testing the impact of any algorithmic changes in your own tool. So if you're improving your tool's phenotypic similarity scoring, you can benchmark the old version against the new version and quantify any gains or regressions in that. You can also test the effects of updates to any databases or ontologies, for example, how the changes in the human phenotype ontology impact gene ranking outcomes. And this can be particularly important as these resources evolve quite frequently. And then we can also look at um, how we incorporate different model organism data or protein-protein interaction data into it, how this affects the performance or how well tools deal with these challenging variant types like structural variants or non-coding mutations. So the key thing that I want to highlight here is that whatever your question is about your algorithm or different algorithms out there in the space right now, PhEval provides the structure and the metrics to explore it. So I can show you a practical application of how we use PhEval. So this is what we did for our paper. So we ran a benchmark of 7 different prioritization tools across 2 separate packet corpora. So we used the phenopacket store, and then we also used a set of structural variant phenopacket. And these were real-world patient-like cases that were extracted from the literature with known causal variants recorded in the phenopacket. So thanks to the standardized input-output structure, we were able to easily compare the results across tools. And we looked at all the different metrics, like how many appeared in the top N entity and accuracy, precision, and recall. And then we had these several plots that could to summarize the findings. So this kind of comparative benchmarking is incredibly difficult without a framework like PhEval, and it's vital for understanding how different tools behave in varied clinical contexts. Simple view of how so it operates in these 3 different phases, which are the data preparation phase, the runner phase, and the analysis phase. So in the data preparation phase, this could be anything that you need to set up for your tool. So this could be setting up the test data like the phenopacket store, or it could be anything you want to do like setting up the database or making any changes to the database. It can all be done in this phase. So in this case, what I've demonstrated here is that we've taken the phenopacket store and we're just kind of curating some VCF that will go along with them. Next, we have the runner phase. This is where we have this modular aspect of PhEval where the runners that will handle anything that's specific to the algorithm running. So this would include like actually running the tool and then transforming the output into this like standardized format that PhEval expects for the benchmarking. And then finally, we enter the analysis phase, and this is where these standardized results are passed and the metrics are computed. So we get the case-by-case performance comparison report that I mentioned earlier, and some publication-ready plots, and then the statistical summary reports. So this architecture was built to like help enhance the reproducibility and the scalability. So whether you're testing one tool or 20, it makes the process a lot easier. So this is kind of a more in-depth way of looking at how this is split up. So this is like a very modeled view, but it can be split up in the data preparation phase here. Then we have the runner and the analysis. So you can see we have the overlap, which will just represent where the data is flowing from one view to the other. So going even deeper into the data preparation phase, say here we have the phenopacket store. This is just an example of what we did for the paper. We prepared the corpus. This was a PhEval utility command, so it's automating another step. So it's checking for incomplete records. So this could be, it's checking whether a phenopacket has the actual variant recorded in the phenopacket, which we later need for the benchmarking, if it has all the gene identifiers, whether it has a complete set of phenotypic terms or if they're all just excluded. It will help filter out so we have that kind of perfect set we need for running the tool. And then we can also update any of the gene identifiers as symbols, so we can change it to a different namespace, or we can update the symbols, say if the HNC is updated. And then the other step we have is like we have a template VCF. So we just use a healthy patient exome from genome in a bottle. We take that and we take the variant from the patient and we spike it into the template VCF just so that we have a way of assessing the tool's way of handling that genotypic data. So the data preparation phase is crucial. So it's validating each phenopacket to ensure its completeness. Then you can see we move into the runner phase. So we've got the prepared corpus from the data preparation phase. And then we move into the actual runner. So we have tool installation and data dependencies. So this is what we call the input diagram. So an example here would be the Xamizer genotype and phenotype databases and the client distribution. And these are kind of combined together to produce these tool commands. So this is how the actual algorithm handles or how you run it. So with Xamizer, we end up with a batch script, which will have all of the samples and how we're running it against each database. And this, with the actual prepared corpus, is used to execute the tool. So this is just running Xamizer. And as our output, we get the raw results. And this is the output directory that we call in PhEval. So for Xamizer, this is just the Xamizer JSON results. And we process this in a way with PhEval helper methods to go from a Polars data frame to then turn it into a Parquet form. The reason we've done this is that Parquet is a compressible format, so it takes up a lot less space. Xamizer and other tools, like when you run it without the variants, it does come up with a large result size, like the file size. So when we compress it into Parquet, it just helps it keep it more containable and can be transferred a bit easier. So then from this, we move into the data analysis phase. So we've got the Parquet files from the runner phase. So we have the standardized results that are also in the output directory. And we run it through the benchmark command. So from this, what we get are these publication ready plots. So the hope is that you can just run the benchmark command and they can be easily plugged in to the paper that you're writing up as is. And then you also get a DuckDB database. And with this, you get a range of outputs. So you get the ranking statistics, you get the binary classification statistics. And then you also get the ROC and precision recall metrics. So this will help with replicating any plots that you need to modify. So you may have decided you want to change the title of the plots. You can just run it through another command in PhEval and it will just regenerate the plots for you. And then we have the performance comparisons, and this is the case by case. So we want to keep everything contained in this DuckDB database. So in a way, if you need to transfer, if you want to share it with another team or someone else to share how the performance is, it's just easily done with just one simple file. So PhEval, I've spoken a bit about the plugin. It's built around the plugin based architecture, which is arguably one of its biggest strengths. So each tool you benchmark is added as a plugin or what we call a runner. So this encapsulates all the specific logic needed to run that tool. So this includes the database access, the command line arguments, the output passing, how things need to be set up within the input directory. This is all contained within the runner. So this separation means that PhEval itself it remains clean and modular, and you can support a new just by writing another plugin without modifying the core database code base. And hopefully this also encourages community contributions. So if there's a team out there that develops a new prioritization algorithm, you can just create and share a runner for it. And this allows other people in the research space to evaluate it side by side with established tools like Xmiser or Gado. So within the PhEval documentation, we have instructions on how to develop your own one. And then there's also a list of ones that have already been implemented. So you can see we've got Xmiser, Lyrical, AI Marvel, and a numerous amount in there already implemented. So this is just kind of a pre-look of how we run a runner. So this is not enforced to like the directory structure, it's just how we kind of how we suggest it should be laid out just for clarity. So each runner is responsible for locating its input data. So under configurations, this is like the input directory. So here we can see I've got the configurations for the Xmiser and the Gado. So this just includes the actual location of the databases. And then this config.yaml is what we have in every single one of the runners. So for this, it's just set out by PhEval, and it helps us kind of document and link what's actually been run. So whether we're running like gene variant or disease prioritization, the name of the tool, and the version. So just helping us have that documentation trail. Then we have the phenopacket corporate location. So this is the test data directory in PhEval. So for this, I'm just showing you a test on 5 phenopackets. And then where the outputs of the PhEval run command will be stored. So this is the output directory. So this is Xmiser and Gado, which is what I'll be showing you. So if we keep everything separated like this, it just helps us be able to trace back any potential errors a lot easier. Everything is separated by tool by configuration. So with the interest of like loads of errors that happen when you do live coding, I've just prepared videos showing you how things work with the runner. So this is me just running the Xmiser runner. And as a side note, all of the runners that we have implemented right now are released on PyPy. So we recommend kind of using it in its own environment for each runner to kind of reduce the dependency conflicts that may occur between different tools. So here I'm just showing you where I am within the directory structure, and you can see this is the same one that I've shown in the previous slide. And then we can check the PhEval installation by just typing in PhEval, and you'll get the commands that we have at the minute. And this is the main PhEval run command that we have. And then there's also all of the arguments that we need to be able to run it, and then the descriptions of what each one actually is. So we pass it the input directory. So this is where like the Xmiser installation is, the Xmiser databases are. And this is consumed by the actual runner interface, and it will know where everything is. The test data directory is just where the phenopackets are. And then we specify the runner. So this is unique to each different runner that's made. So it should be a different name, and this helps PhEval identify which one it should actually run. And the output directory is just where we want to spit out the results. So this will be both the raw results and the PhEval standardized results. And then we can also specify which version of the tool we're actually using. And then we enter the command and everything should run. And you'll get a combination of both like the phenopacket log and the tool log. So it should be super helpful with debugging any issues, whether it happens internally with PhEval or the actual tool that you're running. And one thing to note is that the time it takes for a full run to complete it is largely dependent on how long it takes for the actual tool to complete it. So you may find some will run a lot quicker than the others. And just a quick note, Xmizer here is being run with just the phenotype, so we're not using any of the VCFs. And then in the end, we post-process the results. So this is happening again by PhEval. So it looks in each phenopacket to see what the correct gene or variant is and labels it. And you can see it's finished. And PhEval should tell you how long it takes just to run those samples. And we could check. We have this is from PhEval. We've got some results in the raw results, these are the Xmizer JSON results, and then we can check to see if they've been post-processed. So then we have the Parquet files. And then PhEval also spits out like a results.yaml file, and this is just some like metadata of the run. So it's the timestamp, what corpus was used, tool and the tool version, and the configuration. So this helps with just tracing back what we need to see. And that's that. And then we have the Gado runner. So it's going to run very similarly to the Xmizer one, but you can see I've used a different environment just for this one. And here we can see that we've got the under Xmizer the results. So we know that the past ones run successfully and we don't have anything for Gado yet. And we could just look into the results to make sure we have all the files. And then we just run the same command, PhEval run, but for Gado. So for the input directory, would give it the Gado installation directory. And then the test data directory would be the same as Xmizer because we're looking at the same data set. We're just running different configurations on the same test data so that they can be comparable. And then the runner is going to be also specific to Gado, and the output directory will be to where we want the Gado results to be. So this should be separate to the Xmizer location. And then we run the tool. You can see it runs a little bit faster than an Xmizer, and it's about 5 seconds. And then we can check whether all the results have actually been generated and if they're in the right place, and if we've got everything that we expect. So we can see that PhEval's created these subdirectories. And then we've got the raw results from Gado, and then the process result of PhEval. And then we've also got the metadata. And then I think here I'm just going to show you what everything looks like when it's all run together. We have all of the results for Xmize and Gado and all of the input commands that are used to actually run it. So that way you can see what has actually been run specific to each tool. So what do the PhEval standardized results actually look like? So this is just how it is. It's very simplistic. We ask for the bare minimum for each tool, just so that it can be extended to tools. Say like Xmizer reports a wide range of different things about the gene variant disease, but then we've got Phentogene or Gado that would just give it a gene symbol and a score. So we're trying to keep it simplistic so it can be accommodated for each tool. So what we have is the only things that we need from the tool, say a score, gene symbol, gene identifier, or it would be a score and a variant or a score and disease. And then what PhEval does, it will re-rank everything. So ignore the rank given from the tool, because what we find is that different tools will rank things in different ways. And this makes it particularly difficult when we want to compare different tools together. So the way PhEval does it is it discounts for the same ranking. As you can see here, it's a 6, and this is because there's like 6 at the same rank. So it should, it's increasing the amount of results that we need to actually look at. And then another thing that PhEval does is that within this parquet, it will label what we know in the pheno packet to be the true positive. So what this GCDH gene symbol is what is actually in the pheno packet as the known diagnosis. So we label this as true and the rest is false. And this just helps us with quickening and making the benchmarking a lot more streamlined and a lot faster for people to run. So to walk you through setting up a benchmark is super simple in a sense that all you need is to set up this YAML configuration file. And this is kind of how it looks when we look into it. So what we do begin with is we give a unique name for the benchmark, and this is just used to label all the output files of it. So we'll use to label the DuckDB database, or we use to label the plots, just so you know that everything has come from this specific benchmark. And then each entry under this runs is a configuration or tool that you're evaluating. So you could be evaluating 5 different versions of Xmize and you'd have 5 different entries under this. You have this Xmize against Gado with the examples that I just showed you in the previous slides. And we give each of these an identifier for the tool or the configuration being evaluated in the benchmark. And this just allows us to name the tools and the plots and the actual identify what you're looking at quickly. So here we provide the output directory, and this is the directory that is ultimately passed in the PhEval run command. So it's the directory that contains these PhEval standardized results. So it allows us to look into finding those Parquet files, finding the true positives labeled in the file to run the benchmarking. Then we have the phenopacket directory. So this isn't actually used within the benchmarking process. It's just there for reference, but it allows us to actually trace back and see what configuration did we run on what test data. So we can trace it back and we have that documentation there for our own reference and for others. And then these are just flags to specify which type of analysis to benchmark. So PhEval supports looking at like gene prioritization, variant prioritization, and disease prioritization. So some tools like Xmizer can support all 3, but Gado can only support gene analysis. And then again, if we were to run Xmizer with the VCFs and Gado without the VCFs, it wouldn't be a fair comparison because Xmizer has that extra genotypic data to leverage. It's not really a fair comparison against Gado that just uses the phenotypic data. So in this case, we just did Xmizer with the phenotypes only, so we can compare it to Gado. And then additionally, you can specify like a threshold for the score. So we could say we only want results that have a score of like 0.5 or above or 0.5 on the score order, whether lower scores are better or higher scores are better. So this could be useful if you want to navigate, look at whether you should include a threshold in your algorithm. And then we move into this plot customization. So this is split into gene plots, disease plots, and variant plots. So the plot type, this can specify the plots to generate. So it's set default as like a cumulative bar graph, but it could be a stacked, could be non-cumulative graph. And then this is where we have the customization with the plot titles. So we can include some like titles with a plot, so it can be plugged into to a paper. And this is optional. So if you leave it blank, there won't be a title there. So we're a benchmark. This should hopefully load. So this is just in the main PhEval base environment. You can see we're in the same directories we have. I've just added this benchmark directory where I have the YAML file and we have all the results. So if we look at the benchmark.yaml, this is the same one I've shown you in the previous slide. So this is what we use to run PhEval. And it's just a simple command, PhEval utils, and it will come up with all the utility commands you can have. And there we have at the top is benchmark. So that's what we use to run the full benchmarking process. And you can see the only thing that we need to provide is the YAML file. and this will produce logs. So hopefully be helpful if there's a debugging issue. Tell you how long it takes. It takes around 4 seconds, and you can say we have the DuckDB and the SVG files are just the plots. So this is an overview of the results of what we've just run, or what I've run and prepared. So what we can see is that Xmiser outperforms Gado, so it captures twice as many of these causative genes in the top ranks. But both of these tools, you can see in this precision recall, they have a very low precision, and this is commonly seen with tools or configurations that don't leverage genotypic data. So what you see, is that usually like Xmiser, when we use genotypic data, the kind of curve is flipped, so it's more like a flipped ROC curve. But what's important here is that you can see, like, if a benchmarking study only reported, say, these metrics, it would make everything look great. You know, you can see Xmiser has found all of them. It's got like 80% in the top 10, and it's got great precision and great ROC curve. But then when you look at the precision is low. So it's not giving you the full picture, but with PhEval, we've like given everything. So it should be super easy to see things that the tools will lack with, or it's not performing as well with in terms of metrics. So when we look at the database, this is just in PyCharm to show you how it would look. So we have these 3 tables. So this first one, the gene rank changes, that's what will give you the case-by-case comparison report. Then this middle table, this is the metrics for like calculating the ROC curves and the precision recall curves. This isn't to interpret, but this is just there so that if you need to generate anything again, any plots again, you can just pass it this DuckDB, the database, in a PhEval command, and it will just generate everything again with the benchmarking YAML file. And then this final one is this gene summary, and this provides you the ranking and binary classification statistics. When we look at the case-by-case report, this is what it shows. It has this Parquet. It would have each case like the Parquet file. Some cases might have more than one gene or more than one variant. It would just be replicated and it would have a different entity here. So here we can identify like, oh, Xmiser and Gado both gave the gene at the top top rank spot for this case. But then here we can see that there's quite a big difference. So like, there's a rank change of 100 between Xmiser and Gado. And this is what's particularly useful for when you want to look at your own tools and see, like, when you make small changes, like where the smaller changes actually happen in each case. Like, is your tool better at prioritizing, say, neurological cases to musculoskeletal or things like that? And then this is just super high level view of some of the results. So this is me just trying to emphasize that there's different views that we can take it, like ranking, classification, the overall performance or error analysis. So like we can see that Xmiser like sort of twice as many as Gado in the top one, and the mean reciprocal rank is a lot higher than Gado. And then we could look at precision F1 score, which are equal for both, and accuracy is equal. We've got Matthew's correlation coefficient, which is Xmiser's W is good at, and the false positive and the false that are represented in that summary table. So there's a lot of things that you can look at for your tool to see like how the varies. And to finish, I just want to and thanking everyone that's been a part of PhEval. Like it wouldn't be possible without all of the everyone that's been involved, Jules, Damien, Nico, James, and Vinicius, and then all of the labs that have also been involved, Peter Robertson and Chris Mungle and Melissa Handel, and David Somi Sutherland, who have been great with helping with different kind of like parts of PhEval. And lastly, thank you to everyone for listening and thank you for having your time. And I'm happy to take any questions.
This was excellent. Thank you very, very much, Yasmin. This was super good. Let's start with see whether someone wants to ask a question. I want to tell a short story that fits with your presentation, but let's see whether others want to go first. All right, since no one raises their hand right now, I also want to kind of tell the story of how PhEval is helping us right now to debug a situation where so it's something that Damien noticed in when he was preparing the next version of Exomizer, basically that the latest version of the cross species mappings for especially HP to MP that made it into the Exomizer database caused a drop in performance for some of the cases that Exomizer was looking at. And this kind of framework really allows us now to do a really cool cycle of work, which is we can. So I basically, he submitted this issue to me. I would go and provide kind of like try to develop a fix in the ontology, generate an intermediate kind of developmental snapshot of the ontology, and he could immediately basically rerun the benchmark using the updated files derived from that ontology. So where these mappings, which were an oversight in the structure of the ontology, actually a mistake in the structure, were fixed. So now we are waiting and seeing whether basically whether this fix that I provided to the ontology actually will trickle through to the performance of the diagnostic tool, which is Exomizer, by looking at basically a benchmark result from PhEval. So this is kind of another view that Yasmin also was pointing out before, that it's not just, from the perspective of let's compare the performance of these algorithms, but really also maybe this can help us identifying gaps in the ontologies that we are building, problems with the way that we are representing information in these ontologies. And sort of that was sort of the vision also in the beginning, that we could do things like that. And I'm very happy that this is now, that we have gone full circle with this, and that's really possible to do now in a relatively straightforward manner. So yeah, and hopefully someone does anyone else have any kind of things to add, maybe something else to say or something to ask?
Yeah, just to reiterate what you're saying, Nico, I mean, it's so much nicer when we change things in the Exomizer software or the database. I feel it's so much more secure now that we can kind of test it properly and release. And when we release, we've not broken things. And if we do break
Yes.
things, we can fix them very quickly. So yeah.
That's very good. Yeah, this tutorial was very well designed. I have to say, very nicely done. So people will definitely come back to your presentation and see when they are setting up their own benchmark. I see some of you, the people in the audience, are actually have to and will love to set up their own PhEval benchmarks as part of their work. So looking forward to seeing how they, use your tutorial here as a support for doing so. And yeah, that's it then. Maybe let's wait a few more minutes to see if someone else wants to ask anything. While we are waiting, maybe is there is there anyone here in the audience that is actually working right now on setting up a PhEval benchmark? I know that James is building one, that he's not here right now, but is there anyone else that is building one?
Yeah, I am. Well, I will be soon. I had some older versions of PhEval that I was using, and it's been a few months since I've ran the analysis. So the tutorial will be really helpful for me, like probably later today or later in the week.
Awesome. Very good, thank you, Aaron. Well, want to push pressure anyone? Thank you very much, Yasmin, for this really excellent tutorial, and we're looking forward for the recording soon. And for everyone else, this is a seminar as part of a series of 3 seminars about variant prioritization algorithms, and we will see Yasmin again for an introduction into how to build your own PhEval runner in, I think, like 2 months time or 3. I forgot, in June, I think. Yeah, so you will hear from us about the remaining presentations and when they're happening in the next weeks as well. So yeah, thanks all for being here, and talk to you next time.